It's so wonderful to be here again for another fantastic President's Pick session on the Heart2Heart Stage at ACC 22 in Washington, D.C. And I'm joined, this is such a special moment for me. I have to tell you, I'm joined this morning by TAVR pioneer, Professor Alain Cribier. Each year, the ACC president has the great privilege of recognizing an individual who has made an outstanding contribution to the field of cardiology and or the broader field of medicine with the presidential citation. This year, I selected Dr. Cribier to receive this honor in recognition of the 20th anniversary of the first clinical TAVR procedure. Dr. Cribier is a pioneering interventional cardiologist and past chief of cardiology at the Hospital Charles Nicolle at the University of Rouen, France. And he serves as professor emeritus and co-director of the Rouen Medical Training Center. He's widely recognized as having performed the first balloon valvuloplasty in 1985, the first mitral commissurotomy in 1995, and he performed the world's first transcatheter aortic valve implantation in April of 2002. He has published more than 1,500 journal articles and received numerous degrees, awards, and recognition for his lifelong dedication and contributions to interventional cardiology. So innovation and collaboration really have been critical themes for me during my presidential year, and we cannot achieve that mission to transform cardiovascular care and improve heart health without either one. His leadership and contribution in moving TAVR from concept to reality is a prime example of just how far innovation and multidisciplinary collaboration and teamwork can go hand in hand to deliver far-reaching and revolutionary changes in how care is delivered. So it's my tremendous honor to welcome Dr. Alain Cribier here today.
It's my pleasure and an honor for me to be with you today. Thank you very much. Thank you so much for the invitation.
Absolutely. So glad that you made the journey. And, I know what the audience wants to know. How did you do it?
I did what? The valve?
Yes. You tell us about the journey that led you to the first implantation.
Well, do we have the full day? Because it will not be enough.
We don't have the full day. You can give me a short version.
I will make it short. Yes, actually, it's a journey. It's like a TV series in two seasons. There was a first season in the mid-80s, when I was really planning to find an alternative to surgery in those patients who could not be operated on because they were too old. And at that time, in the mid-80s, too old meant 70 years of age. So we were losing a lot of patients, without being able to do anything. So I started thinking about dilating the valve. So this was the first season. Balloon aortic valvuloplasty per se was a big story as well. The first case was followed by a short series of patients published in The Lancet. And immediately after that, the balloon aortic valvuloplasty spread in the world like crazy. Tens of thousands of patients were dilated, and they had absolutely no way to control the indications. So people did absolutely what they wanted. The dilated patients were excellent surgical candidates and so on. And after 6 years, there was a first publication of a big series of patients done in the United States showing that there was early restenosis. So then I was very disappointed, like many others, and I had the choice of giving up or moving to season 2.
Season 2. Season
2 was the development of transcatheter aortic valve. So this is when everything started, in 92, I started thinking about putting a stented valve because the Cordis had developed at that time a peripheral stent which was 23 millimeter in diameter. So I think that maybe we could use this kind of stent, implant the valve inside, and actually try to do a percutaneous aortic valve replacement at the time of balloon valvuloplasty. So balloon valvuloplasty followed by implantation of the valves. This was my idea. But it had to be confirmed, and we did in Rouen with my colleague, Alain Cribier, who is now director of the Department of Cardiology after me. We did a landmark anatomic study on cadavers of patients who died of aortic stenosis, and I could prove that it was feasible against the opinion of everyone, all the surgeons, of course, and also the cardiologists say that if you put a stent in the aortic valve, you will occlude the coronary arteries, destroy the mitral valve, you will have a permanent block, endocarditis, the result will not be good, and the valve will migrate in the brain. So it was very discouraging, but I could prove that it was really feasible. And so I came with a dimension of the valve, 17 millimeter in height and 23 millimeter in diameter, showing that we had enough room to implant a valve without occluding the coronary arteries and mitral valve. So this was the first step. Absolutely, it's a landmark trial.
Okay, so wait, before you go on, because this is so fascinating, tell me from the thought that you had that this was something that you wanted to do to the point where you got to this point. How much time are we talking about?
We are talking about, I started doing this valve implant, this anatomical trial in '94. And then very rapidly, actually, I find on my way somebody who is not here, unfortunately, but an engineer from Johnson & Johnson in a meeting in Paris, and Stan Rabinovich, who pay attention to my concept of valve implantation, and he was, I must say, he was the only one interested by the topic. So immediately he went back to Johnson & Johnson and discussed with Stan Rowe, who is here, and all together, they motivated themselves and they said, well, this is a great idea, we have to do something. So it took again 5 years, to get to the...
So were you at a meeting like this when you talked about that idea and the engineer just heard you? So, I mean, what I'm trying to find out, and for those innovators out in the audience, was it luck or was it intentional?
You need luck.
Okay.
You need luck. And actually, Mark Zuckerberg said, the father of Facebook said that when you have some good idea, if you want to innovate in whatever the field, not only in medicine, you have to work hard, you have to spend time, you have to convince yourself and so on, all these parameters, but also you have to have luck with you. And I had luck with me. And the luck came when I met Stan Rabinovich and Stan Rowe, and this was a big luck for me. So this was in 96.
Well, you can call it luck, or you can say, unless you put your idea out, nobody's going to know, and maybe it will be something that resonates.
Yeah.
And then you do have to get a little lucky, but I think it's about your idea.
Yes, well, actually, yeah, I was a little responsible of it. And
right.
well, actually, if I can just continue briefly, 2 years to meet people, to discuss whether a protocol, a research program with Johnson & Johnson. Also, Stan and Rabinovich and Stan Rowe discussed with Martin Leon, who was at that time medical director of Johnson & Johnson. And he was also very much interested in the idea and the concept. And he said, well, there is something to do, there is a potential, in implanting a valve, even though it was a little crazy. It was considered a little crazy. It was not enthusiastically accepted. But, well, we should do something. And so we had the first research program with Johnson & Johnson, which failed totally, so they did absolutely nothing. And it's only in '99 that we decided, together with Martin Leon, Stan Rowe, and Stan Rabinovich, to found a startup, percutaneous technology. And I have to give this message to those people who want to innovate in medicine. Don't hesitate to found a startup, if you want to develop your idea, because getting through big companies, you are wasting time. People are they are occupied to do many things. They are not paying enough attention to your project. So if you create a startup, you have to find the money. And so this is something that my partner did very well, to find the first investors and the first company interested in putting the idea into a device.
So your advice is, if you have a great idea, don't hesitate to start.
No. I think it's still true now. It was in '99, but I think this is a good message. And so after that, finding support, you have to get a partnership because you cannot do things alone. So finding a good partnership is essential. And you have to find an industrial partnership, and you have to find the finances, the money to make it.
Right.
And so fortunately, and thanks to Martin Leon and my partners, they found a small company in Israel who were the first investor and also who gave the money to start making the device. So, and they made a prototype, and it took, I would say, just 6 months, to make a prototype corresponding exactly to my drawing for the patent, that I had. And so a stented valve looked good. And so we decided to start big work in the laboratory in Israel. And maybe 6 months, 1 year later, we started the preclinical animal experiment in Paris on the animal model, on the sheep model.
So you used the prototype on the animals?
Yes, yes. And, for almost, we are changing the prototype almost for each animal, and we did 100 animals, so we are making
100.
progress, we improved the delivery system, we try to understand absolutely everything concerning valve implantation through catheter, balloon expandable valve. Unfortunately, these animals are stupid because they don't have calcifications on the valve. They have healthy valves, and also they have the coronary arteries and the mitral valve very close to the valve itself. So it means that we could not very efficiently schedule the orthotopic implantation of the aortic valve inside the native valve of the animal. So we are a little disappointed by that. But finally, we find everything else. We develop with Alain, and we were concerned to find the solution to all the problems that we could have, to push the device inside the sheath, 24 French sheaths introduced in the carotid artery and put it in the descending. We did something extraordinary with my friend Hélène. We developed a model for chronic implantation of the valve in the systemic circulation in the descending aorta. It looks like crazy, but if you want to do a chronic experiment, you have to put the valve in the systemic system. And unfortunately, if you put a valve in descending aorta, there is no gradient, so the valve is dysfunctioning after a few hours. So we have to create a gradient by creating an aortic insufficiency, removing some pieces of the native aortic valve with a biotome, and so we could follow the valve 5 months, and we did that in several animals, enough to convince the FDA that this is exactly what they want, to prove that the valve is working.
So I'm going to stop you. So 100 animals that you tried the prototype on.
Yeah, sure.
And you continue to refine the...
Yes, every time.
Every time. And tell me about the timeline on that. How long was that?
Not that long, actually, between 92 and 2002. So it's 3 years. Yeah.
Okay.
2 years and a half, 3 years.
And you took that data and you went to the FDA.
Yes. Yes, we showed to the FDA the chronic experiment.
And tell us about that experience. What was that like?
The experience of what?
Going to the FDA, and how did they receive you and all of that?
Well, they were not very enthusiastic, I must say, but they did appreciate that we did this work, and so the door was opened to further implantation in humans, if ever it was working in humans. So we had to demonstrate also that it was working in humans, which was a big step. A very big step.
Okay, so tell us that story. We're all wanting to know.
Well, after 3 years of animal experiment, we were almost ready to start in human, because we knew that there was a limitation in the animal experiment because the animal had no calcium, and so we could not schedule, we could not foresee what would happen in valve implantation in human. We just got all the techniques, to place the valve and to manipulate the valve in the systemic circulation, that's it. So once we had to move to a patient, and when you have a startup and when you have been spending time and money, to make a prototype, you are hoping to have a good patient to start with. An ideal patient, with no issues, no special issues. And actually, it's exactly the opposite that happened. I received a patient from the north of France. It was unbelievable. This patient was very young. He was 57 years of age. Oh. And he was in cardiogenic shock with no ejection fraction. The ejection fraction was 10%. He had intraventricular thrombus, floating, and he had no femoral access. So, of course, the device had been thought to be implanted through the femoral artery, retrograde,
And now
always.
you couldn't do that.
And two occluded femoral bypass and a subacute ischemia of the leg, because one of the second bypass was occluding. And so we had these patients, and he was sent to us not for a valve, because the valve did not exist, but for dilating the valve in emergency using the transeptal approach. That we had a good experience of it with mitral commissuratome, and we did many balloon aortic valvuloplasty using the transeptal approach in patients who had no arterial femoral access. And so I did it, transeptal, and it didn't work. The patient had several episodes of cardiac arrest, and so I had to stop it. And but the patient did survive, and 1 day later he was back in cardiogenic shock. When I say cardiogenic shock, it was really cardiogenic shock. Under vasopressive drugs, he had only 70 millimeter of mercury of aortic pressure.
But this was your first patient.
Yes, the first patient. It was horrible. It was the worst. So. After the failed balloon aortic valvuloplasty, I had to take a decision of moving to the first valve implantation for saving his life, because he was 57. Mm-hmm. If he had been 75, we won't be there today, probably. But he was 57, so I sent a long message to my partner of percutaneous valve technology saying, we have this patient, he's dying, he has no femoral approach. He has a thrombus in the left ventricle. What do you think? I would like to make it, but it's very risky, and especially for our startup, So, and they said yes. They said yes, and I thank them very much for that, because there was almost no chance of success, but it was the only possibility of saving the patient's life. And this is for ethical reasons, actually, that they said yes, you can go, and you can move on. So we did it.
But tell me about the conversation you had with the family.
It was interesting because, I couldn't sleep at night. No, it's absolutely unbelievable. I couldn't sleep at night because, when you have to explain to a patient that he will be the next sheep, actually.
Yes, exactly. That's why I was asking you.
It's a little difficult. So I didn't sleep at night. I was trying to find the right words and so on. And I went to them and I said, we are developing a new technology that might be useful for you, but we have no experience in human. And the guy, there was the gender of the patient who was here, and he stopped me and said, well, stop your history. I am the first patient who received a laser on the coronary artery in Germany, and it was amazing. He said, well, this is something that you have to do. And the patient was, I want my life to be saved. It was great. So finally, we did it. And we asked a valve to come from Israel during the weekend, because we didn't have any valve in the laboratory. So we asked a valve, and the people from the company came with the valve to our lab, and we did that. And I must say, without any problem.
Wow.
It's absolutely.
How long did it take you?
It took 45 minutes, I think. 1 hour, I would say 1 hour. We did the transeptal approach, no problem at all. I was not sure, because never it had been done before, cross the septum with a valve. We didn't have any delivery system. It was just a valve crimped over a balloon, so the diameter of the whole thing was around 9 millimeters. So I dilated the septum with a 10 millimeter balloon, that's all. And we had some resistance, but we could go through the septum. We turned around the floating thrombus in the left ventricle, cautiously. And we moved up the valve to the aortic valve. And then, it was difficult to... the gadwall was exiting from the contralateral femoral artery, so we had a good rail, to push the valve. This was full improvisation. and unbelievably, you know, I could place the valve right in the middle of the calcium. And it was a bicuspid valve, actually. I forgot to tell you, this is
Wow.
the cherry on the cake. You know, it was a type 1 bicuspid valve, very calcific. And so we had absolutely no experience of that, of course. And so I didn't know exactly where to place the valve. I placed it right in the middle of calcium, inflate the balloon, and immediately, I don't remember whether Stan, you were there? No, no, you were not there. But I described the whole thing. But immediately, you know, I could see the patient, the color on the face of the patient coming back, you know, from gray to pink. Immediately, the patient was smiling. We stopped the vasopressors. Aortic pressure
Wow.
it was unbelievable. The heart was beating normally. It was just tremendous. And so. With my colleague Hélène Elchaninov, we did this case with her and another guy, Christophe Tron, and we were 3 around the patient to do that. And we had decided that if the valve was working 30 minutes, we would say that it's a success.
Oh, so you waited for 30
So,
minutes to declare success.
We were watching the time, you know. 15 minutes, 30 minutes, 1 hour, 2 hours, 3 hours. The patient was back in his room and he was sitting up. And in the afternoon, we drank champagne with him in his room. You know, it was absolutely a miracle. A miracle.
That's amazing.
Yeah.
How did you handle what you do about the anticoagulation? You said that there was a thrombus.
Well, actually, we had done that in the animal. I decided myself, I must say, that we will have to give aspirin and clopidogrel. And so we did that in the animals, and for the chronic experiment, it was working well. So we did the same for the patient, and actually, it's still done today. It's exactly what we did. I didn't want to give anticoagulant because I was afraid of a bleeding complication and so on. And it worked out very well. So actually, this was a kind of historical event. I realized that something could be done in this field, that I was not totally mad, you know, and I went to my car and I cried.
Oh.
I swear it's true, you know, it was very emotional, you know, people were crying in the room around, it was unbelievable. And then one day after, the patients received all the journalists from the TV and the newspapers, and he gave interview during 1 day. Unbelievable.
Is he still alive?
No, unfortunately, because 4 months later, the valve was working well, but he had an ischemia of the leg when he came, and nothing could be done, and we had to go to an amputation of the leg, and he did not recover surgery. So we lost the patients like this, but you know, this patient has a huge responsibility in the next steps. So
I want to ask you, think back, it's 20 years, right? When you reflect back to the 20 years and the journey, and you've been asked to talk about this, would you have changed anything? What did you learn?
No, actually, I think I won't change anything, you know. You can change things if something didn't work, but if you are not satisfied, if you have problem issues, you know, we never had any problem with the valve development. During 20 years, it has been a constant success, constant expansion of the indications, constant improvement of the technology, so nothing to change, just do the same. We do the same, exactly. And, you know, the next step, if I, do I have time to say?
Yes, we have about 6 minutes.
It took to me 1 year to convince the health authorities in France to give us the green light to do a series of patients. Because the experts at the health authorities were surgeons, cardiac surgeons, you know, of course. And so the cardiac surgeon said, never do that, you know, this is absolutely crazy, it will kill all the patients. You know, he was very lucky with the first one, but the next one will be done. And so 1 year later, I get the green light to do a series of patients using the transseptal approach only, because this is the one that had been used for the first patient.
But it took a year after you did a patient successfully.
Yes,
to do that.
1 year later.
Wow.
And the definition of a compassionate case was a patient who is supposed to die within 15 days. No, so I started with this, the same kind of patient, and for the first one, transseptal approach. It was not easy to do, and we did 40 patients, which were reported in two different tissue of the jack, and with a very good result. Actually, it was amazing, and the patient, it's big story, but one of the patients, the number 3, for example, she was dying. She came from Paris. She was dying. She was obese, and we did that transseptal and so on. And 1 year after, she came to the TCT meeting in United States, and she appeared on the stage, to explain to the people how good it was to have a valve implanted. So this was a compassionate case, and the cases that we had were like this. We had several patients who survived several years, and then the second chance is that finally our company was acquired by Edwards Life Sciences, thanks to Mac Moussalem, because I think that your colleagues were not really enthusiastic about the acquisition of this valve in Edwards Life Sciences. But then, with the capability and the engineering that they have in this company, and the association with the Israeli company that they acquired at the same time, the technique was immediately improved very much, and so we had the Edwards Sapien valve that you have heard, and which was a progress, and with a delivery system coming from a femoral artery, and it was absolutely great. John Webb in Canada make some experiment of this retrograde approach very successfully, and so this was dispatched in the world. After that, there was the first in man. I was there in Leipzig with the transapical approach, which was, this was extraordinary, because from 1 year to another, we were covering all the needs, because all the patients who could not have the valve implanted transfemoral, they could have it implanted transapical, and very well, actually. So it was absolutely great. And you know the rest of the story. I don't have to.
Well, no, this is a fascinating discussion, and we'd love to have more time, but we are on a time limit.
Sure.
But I want you to reflect over the 20 years, and there's a lot of budding innovators out there. What are some of the pearls of wisdom that you learn in this journey that you can share with them?
Well, actually, first of all, when you have an idea, it has to correspond to a clinical need. So this is something very important. The first thing is to detect, to have the capability of detecting the unmet clinical needs. This is absolutely essential. And I learned that from Swan and Gans in Cedars-Sinai Medical Center, where I had a fellowship in '77. And they explained to me how important it was not to do something like this to play with, just it has to correspond to clinical needs. Then you have to trust yourself and say, well, I am able to be the one who will solve this issue. And so you have to be a little paranoid. You have to believe in yourself very much. You have to convince yourself, and you have to try to demonstrate that you are not totally foolish, because the first reaction of people when you are talking about an innovation in medicine is to say, this guy is crazy, because if he was not crazy, this innovation would already exist. So if it does not exist, it means that it's impossible to do. So you have to try to think about all what can happen, the technology, the complication, the interest for the patient, and so on. Then you have to to start discussing around you with your partner, your close partner, to have their reaction and try to convince themselves. Then you have to bring scientific data to prove that you are not silly. And this is what I did when we went to the animal lab and to do the preclinical data. And that's it. And after that, I explained to you, you have to be very perseverant, never being discouraged. If you fail, that's not a problem. You have to find a solution to overcome the difficulty. And so, it's a number of parameters.
But do you think you ever envisioned that this is where you would be 20 years later?
No, not at all. No, not at that time. No. And my goal was absolutely to bring an alternative to surgical valve replacement in those patients who were declined for surgery. And then I started, after the first series of compassionate cases, when it was working so well, the patients were improving so much, I started to think that maybe we could think about an expansion of indications. But it could not be done like this. And it went through a randomized trial. And again, Edwards Life Science has been absolutely unbelievable when they sponsored this 3 randomized trial in non-surgical patients, high-risk patients, intermediate-risk patients, and finally low-risk patients. We move step by step. But you must realize that this is an innovation we started by the worst patient, which is very unusual. If you compare with angioplasty, for example, with coronary angioplasty, you have a good idea, a good technology, you believe in it, and you start by the easiest patients, and then progressively you move to more difficult cases and the worst indications. But we were pushed by the health authority to do exactly the opposite, starting with dying patients, demonstrating that this innovative technology can save the life of dying patients, to progressively move to non-surgical candidates, which is absolutely remarkable.
All right, my last question. Looking forward, where do you think we're going?
Far. No, well, actually, we can. No, we have to be conscious, that some patients will remain good surgical candidates, and we are not here, to demonstrate that we can do everything instead of the surgeons. The surgeons are doing a very good job, and we have indications that remains good case for the surgeon. But I think that with the several iterations of the devices that you can observe today, the technique is getting easier and easier. The complications rate are dropping to almost 0. And so the baseline, when you have an aortic stenosis in the next future will be to implant a valve. The only question today is the durability of the valve that we don't know, because the devices are changing every 2 years. We have a new device, so you cannot really conclude that it will work 20 years. We don't know, but we have absolutely no alarm on the durability of this valve in comparison to the bioprosthesis, surgical bioprosthesis. So I think that this technique will continue to develop. The low-risk patients will continue to increase, but that's for the aortic valve. Probably we will find a way to treat the aortic insufficiency as well. It's already done, if I can say, in some patients. But also the importance of this technology has also been to open the field to many other indications of percutaneous valve implantations for other valve disease. Has really pushed the people to work on the structural heart disease indications, and well, I am satisfied.
Well, that's a good note to end on. Thank you so much. Obviously, we could go on, but you truly are a legend, and you have transformed this field.
Thank you so much.
Thank you so much.
Thank you, Mrs. President. Thank you. Thank you.